everybody. I'm Dr. Swathika, Assistant Professor of Biochemistry of Lakshmanarayana Institute of Medical Sciences, Pondicherry. Today, we'll be discussing regarding tricarboxylic acid cycle. It is also called as Krebs cycle. So, shall we move on to the class now? So, basically, what are the functions of citric acid cycle now we'll see. Citric acid cycle or Krebs cycle or tricarboxylic acid cycle, it is the final common oxidative pathway that oxidizes acetyl coenzyme A to carbon dioxide. It is the source of reduced coenzymes that provide the substrate for the respiratory chain. And it also acts as a link between the catabolic and anabolic pathway. Hence, it is also called as an amphibolic pathway. And it provides precursors for the synthesis of amino acids and nucleotides. And the components of the cycle has got a direct or an indirect controlling effect on key enzymes of other metabolic pathways. So here you can see in this slide the sources and utilization of the acetyl coenzyme. Acetyl coenzyme is a starting product for tricarboxylic acid cycle. So from where does this acetyl coenzyme come from? It can come from the breakdown of carbohydrates, fats, and also amino acids. So mainly the glucose will break down, undergo glycolysis to form pyruvate. This pyruvate, which is formed in the cytosol, is shifted into the mitochondria after combining with, by the help of an enzyme, pyruvate dehydrogenase, and then it is forming acetyl coenzyme in the mitochondria. This fatty acids also contributes to acetyl coenzyme. Amino acids, mainly the ketogenic amino acids, will result in formation of acetyl coenzyme. What is the fate of acetyl coenzyme? Acetyl coenzyme will undergo oxidation by this cycle, the Tca cycle, or it will get converted into fatty acids. It will contribute to fatty acid synthesis, or it will be converted into cholesterol. This cholesterol in turn will help in formation of steroid hormones, or this acetyl coenzyme can be converted to ketone bodies. What are the reactions of oxaloacetate? As you can see here, glucose undergo glycolysis to form pyruvate. This pyruvate has got two fates. One is it can get converted into acetyl coenzyme to enter into the Tca cycle, or it can get converted into oxaloacetate. This oxaloacetate undergoes malate shuttle, that is shuttle between conversion of malate to aspartate and aspartate to oxaloacetate in certain reactions or in certain metabolic pathways where oxaloacetate needs to be transported across the mitochondria. And this oxaloacetate can get converted into phosphoryl pyruvate by virtue of gluconeogenesis, and this phosphoryl pyruvate again can get converted into glucose. So this is the entire Tca cycle you can see here. So you can see the starting product, acetyl coenzyme, which is a 2-carbon compound. This 2-carbon compound will combine with the 4-carbon compound, which is nothing but oxaloacetate, with the help of an enzyme, citrate synthase, which is the first enzyme of Tca cycle to form citric acid or citrate. This citric acid will undergo hydrolysis to form a water molecule will be removed from this to form cis-acetate, and this cis-acetate again consumes another water molecule, and this reaction is catalyzed by the enzyme metabo. This is the second enzyme to form isocitrate. This isocitrate is converted into oxalosuccinate with the help of enzyme isocitrate dehydrogenase. Since it is one hydrogen atom is removed from isocitrate, this enzyme is called as dehydrogenase, the isocitrate dehydrogenase. This H plus ion is contributed to NAD plus to form NADH. So one NADH is formed in this third step of Tca cycle. Then the oxaloacetate undergo decarboxylation to form alpha ketoglutarate. So the acetyl coenzyme A, which is contributing 2 carbon atom, among the 2 carbon atom contributed by acetyl coenzyme A, 1 carbon atom is released here as carbon dioxide. Later, this alpha ketoglutarate again undergoes a decarboxylation reaction and reduction reaction with the help of an enzyme alpha ketoglutarate dehydrogenase to form succinyl coenzyme. So the second carbon atom of acetyl coenzyme A will be released here. So the acetyl coenzyme A, which is incorporated into the Tca cycle, is completely oxidized. And again, because there is a reduction reaction happening, this H plus ion removed from the alpha ketoglutarate is contributed to NAD plus to form NADH. Then this succinyl coenzyme will undergo substrate level phosphorylation. That is, the phosphorylation happening at the level of substrate, and it results in formation of 1 GTP. And this coenzyme A is also removed with the help of an enzyme succinate thiokinase to form succinate. This succinate will undergo again a reduction reaction where FADH2 will be formed. The sixth step, FADH2 is formed with the help of an enzyme succinate dehydrogenase. So you can see here, carbon dioxide is removed at the steps 3 and 4, and NADH is formed at the steps 3 and 4, and FADH is formed at the step 6. So then this fumarate will be hydrolyzed. A water molecule will be added with the help of an enzyme fumarase and results in formation of malate. This malate will undergo again a reduction reaction to form oxaloacetate, and again NADH is produced at this 8th step too. So this is the entire Tca cycle. So acetyl coenzyme A, a 2-carbon atom which enters the cycle, are released as carbon dioxide in steps 3 and 4. So alpha ketoglutarate dehydrogenase is a multi-enzyme complex which will result in formation of ultimately NADH and FADH2 by certain step of reactions or a certain cycle of reactions. So this alpha ketoglutarate is converted into hydroxysuccinyl TPP with the help of an enzyme alpha ketoglutarate dehydrogenase, and thymine pyrophosphate acts as a coenzyme, and 1 carbon dioxide is removed here. This hydroxysuccinyl thymine pyrophosphate undergoes succinylation with the help of an enzyme trans-succinylase, and lipomate acts as a cofactor here to form succinyl lipomate. This succinyl lipomate again undergoes trans-succinylation reaction to form succinyl coenzyme A. This succinyl coenzyme A and the another coenzyme A is added to succinyl lipomate, and it results in formation of dihydrolipomate. This dihydrolipomate undergoes dehydrogenation reaction to form lipomate, and in this reaction FADH2 is formed. This FADH2 is converted to FAD and results in formation of NADH. So these are the 5 enzymes which are present in the alpha ketoglutarate dehydrogenase complex. One is alpha ketoglutarate dehydrogenase per se, trans-succinylase, and dihydrolipomate dehydrogenase, and then this reduction reaction. So the cofactors are thymine pyrophosphate, lipomate, coenzyme A, FAD, and NAD. Summary of Krebs cycle or citric acid cycle. So this is the first step where oxaloacetate combines with acetyl coenzyme to form citrate. Citrate is converted into aconitase, aconitase to isocitrate, isocitrate to oxalosuccinate, oxalosuccinate decarboxylated to form alpha ketoglutarate, and when conversion of isocitrate to oxalosuccinate, 1 NADH is formed, and alpha ketoglutarate to succinate coenzyme conversion, 1 NADH and 1 carbon dioxide is removed, and 1 NADH is formed. Succinate coenzyme undergoes substrate level phosphorylation to form succinate. Succinate is again dehydrogenated to form fumarate. Fumarate to malate. Malate again dehydrogenated to form oxaloacetate. So NADH is formed in the steps 3, 4, and 8. And substrate level phosphorylation happens in step 5, and decarboxylation happens in step 3 and 4. So ATP, which is the ultimate product of this oxidation of foodstuffs, will inhibit this conversion. So this is a physiological regulator of citric acid cycle, and pathologically the citrate to isocitrate conversion is inhibited by fluoroacetate, which inhibits the enzyme aconitase. And NADH, if it is present in more when there is energy replenished inside the cell, it will inhibit ADP. It will inhibit this reaction, conversion of isocitrate to oxaloacetate, and ADP, which is which means that there is an energy depletion inside the cell, will physiologically activate this step. And here arsenite will inhibit this enzyme, alpha ketoglutarate dehydrogenase enzyme. hence this conversion is not happening, and malonate will inhibit succinate dehydrogenase enzyme. So this enzyme is called as first enzyme is citric synthase, second enzyme is accommodase, third enzyme is isocitrate dehydrogenase, fourth is alpha ketoglutarate dehydrogenase, fifth is succinate thiokinase, sixth is succinate dehydrogenase, seventh is fumarate, and eighth is malate dehydrogenase. So inhibition is by physiological inhibition is by ATP and ATP, which inhibits this enzyme citric synthase, and NADH, which inhibits the enzyme isocitrate dehydrogenase. Physiological activation is the ADP, which activates the enzyme isocitrate dehydrogenase. NADH generating reaction, isocitrate dehydrogenase. And pathological inhibition is fluoroacetate, which inhibits the enzyme accommodase, and arsenite, which inhibits the enzyme alpha ketoglutarate dehydrogenase, and malonate, which inhibits the enzyme succinate dehydrogenase. And totally 3 NADH is produced in the citric acid cycle. Each NADH will result in formation of 2.5 ATPs. So totally by virtue of NADH production, around 7.5 ATPs are produced, and there is one substrate level phosphorylation happening in the fifth step, which results in formation of one GTP, which contributes to 1 ATP to the ATP pool, and then one FADH2 is produced at the sixth step, which will result in formation of 1.5 ATP. So totally contributing is 10 ATP per cycle of citric acid coenzyme oxidation. So here you can see the ATP generation steps. The step 3, conversion of isocitrate to alpha ketoglutarate resulting in formation of NADH. So 2.5 ATPs per NADH. And fourth step, alpha ketoglutarate to succinate coenzyme 1 NADH is produced again, which contributes to 2.5 ATP. And fifth step, succinate coenzyme to succinate GTP, which results in formation of 1 ATP because of substrate level phosphorylation. And sixth step, succinate to fumarate FADH2 formation, which results in formation of 1.5 ATP. And eighth step, malate to oxaloacetate, which results in formation of 2.5 ATP, totally contributing to 10 ATPs. This is the stoichiometry of Tca cycle. So the starting products is acetyl coenzyme, which combines with oxaloacetate, and the coenzymes FAD, NAD plus, and other cofactors GDP, all contributing to formation of 2 carbon dioxide, oxaloacetate, FADH2, 1 FADH2, 1 GDP, 3 NADH. So Tca cycle is also called as anabolic pathway because Tca cycle is also called as anabolic pathway because it is both catabolic and anabolic, and it is also called as anaplerotic cycle, where there will be catapleresis happening and anapleresis happening. So those intermediates which will deplete the Tca cycle intermediates are called as cataplerotic role, and those intermediates which will replenish the Tca cycle intermediates has got anaplerotic role. So here you can see the effects of Tca cycle intermediates. That is the cataplerotic role. So the acetyl coenzyme combining with oxaloacetate to form citrate to alpha ketoglutarate acid. There is a series of reactions happening here, and this alpha ketoglutarate will result in formation of the neurotransmitter GABA in brain. And similarly, once alpha ketoglutarate is converted to succinate coenzyme, this succinate coenzyme contributes to heme synthesis and also for ketolysis. This succinate coenzyme, when converted into malate, this malate can undergo gluconeogenesis. So this malate is being depleted from Tca cycle for the gluconeogenesis to happen whenever the cell is in need. And this malate, when converted to oxaloacetate, again this oxaloacetate can get converted into the amino acid aspartate. And this acetyl coenzyme, as such, the starting product can go into fatty acid synthesis cycle, and thereby Tca cycle cannot happen. This is how efflux of Tca cycle intermediates happen. Influx, there is an anaplerotic role of certain compounds or metabolic intermediates which will replenish the Tca cycle intermediates. Here you can see pyruvate is converted into acetyl coenzyme and oxaloacetate. This pyruvate is being replenished from glucose and also from tryptophan to alanine, and this alanine will get transaminated to form pyruvate. Similarly, aspartate in reverse can result in formation of oxaloacetate with the help of malate aspartate shuttle. And this acetyl coenzyme combines with oxaloacetate to form citrate to alpha ketoglutarate. This alpha ketoglutarate can be replenished with the help of amino acids. Certain amino acids called as histidine, arginine, proline, and odd chain fatty acids also. All these things will result in formation of glutamate, which will contribute to the formation of alpha ketoglutarate. This alpha ketoglutarate, when converted to succinate coenzyme A. This succinate coenzyme A can be replenished by valine isolation and methionine, which result in formation of protein coenzyme A and succinate coenzyme A. And certain amino acids, aromatic amino acids, vinyl tyrosine, will replenish fumarate. This is how anaplerosis of Tca cycle intermediates happen. And coming to the metabolic defects of oxidative metabolism, there is the Tca cycle. Pyruvate dehydrogenase enzyme, which helps in conversion of pyruvate to acetyl coenzyme A. If it is deficient, there will be lactic acidosis and neurological disorders. And acyl coenzyme A dehydrogenase, which will convert fatty acyl coenzyme A to unsaturated fatty acyl coenzyme A. If this reaction is not happening, there will be organic aciduria, glutaric aciduria, acidosis, hypoglycemia, and so many other manifestations. Pyruvate carboxylase, which helps in conversion of pyruvate to oxaloacetate. So oxaloacetate has to be in adequate amount for Tca cycle to happen without any interference. So if it is not happening, lactic acidosis, hyperammonemia, and hyperammonemia can manifest. How the citric acid cycle or Krebs cycle is regulated. There are four main things which regulate citric acid cycle. One is the citrate and citrate synthase, substrate citrate, and the enzyme citrate synthase, and availability and cellular need of ATP. Third is isocitrate dehydrogenase. Fourth is alpha ketoglutarate dehydrogenase. So citrate and citric citrate synthase. Formation of citrate from oxaloacetate and acetyl coenzyme is an important part of the regulation of citric acid cycle. Here ATP acts as an allosteric inhibitor of citrate synthase, and citrate will allosterically inhibit phosphofructokinase, which is a key enzyme of glycolysis, and stimulates fructose 1,6 phosphatase, a key enzyme of gluconeogenesis, and activates acetyl coenzyme carboxylase, a key enzyme of fatty acid synthesis. So what happens when there is more of citrate? That is, the cell is energy is adequate inside the cell and the cell doesn't need energy. Citrate, which is more formed, so the further inhibition is happening. Further oxidation of citrate is not happening. So there will be more of citrate. This will allosterically inhibit the glycolysis and it will promote gluconeogenesis and fatty acid synthesis. And then second one is the availability and cellular need of ATP. So when the energy charge of the cell is very low, this Tca cycle will operate at a faster rate. And the cycle is tightly coupled to the respiratory chain providing ATP. So in any case of hypoxia or anaerobic conditions, there will be inhibition of ETC when NADH and LPDH2 are accumulated, which in turn will cause inhibition of Tca cycle. The third regulator is isocitrate dehydrogenase. Here ATP acts as a positive modifier, enhancing the binding of substrate, and NADH is an inhibitor. Fourth regulation is at the level of alpha ketoglutarate dehydrogenase. It is inhibited by its further subproduct and product inhibition, acyl coenzyme A and NADH. Inhibitors of Tca cycle. There are main three inhibitors, pathological inhibitors of Tca cycle. And aconitase, which is inhibited by fluoroacetate. Enzyme aconitase, which converts citrate to aconitate, is inhibited by fluoroacetate, which is a non-competitive inhibition. And second one is the alpha ketoglutarate dehydrogenase, where alpha ketoglutarate to succinate coenzyme A conversion is inhibited by arsenate. Again, it is a non-competitive inhibition. This succinate dehydrogenase, where succinate to fumarate is converted, is again inhibited by malonate, which is a competitive inhibition.